So we are back for our next session, the research section. Really excited to hear all the great research that's been going on. And we're going to start with Erin Allen and Jack Giroux, and both of them with Georgian Bay Forever. We heard a little bit from Curtis about the really nice partnerships going on there, and we're going to learn more about their drone imagery capture. So really excited to hear about the work that you've been doing. Thanks for joining us today.
Oh, thank you so much for having us.
Yeah, thank you.
Just get my screen up for everyone to see. There we go, so you can all see me okay?
Can't see the screen just yet.
Oh.
Nothing yet.
Interesting.
Oh, looks like something's loading. That's perfect.
Okay, awesome. Thank you. Okay, so hello everybody. I'm Erin Allen. I'm the Conservation and Restoration Team Lead at Georgian Bay Forever, and I'm here with Jack Giroux, our GIS Technician. And we're here to talk to you all about how we're using drones to monitor invasive Phragmites in Georgian Bay. So since purchasing our drone in 2023, we've been working to develop effective and consistent practices for deploying our drone in the field, and thanks to Jack, a method for identifying Phragmites from the images that we collect. So thank you all for listening, and thank you to everyone at the Ontario Invasive Plant Council for hosting this. Trying to get my slides to click for me. There we go. So Georgian Bay Forever, I'll start off with who we are. Georgian Bay Forever is a charity dedicated to scientific research and public education on Georgian Bay's aquatic ecosystems. Our mission is to protect, enhance, and restore the aquatic ecosystems of Georgian Bay for future generations by taking action and funding accredited research on water levels, water quality, and ecosystems. So in terms of Phragmites management, we've been working to remove frag from the coasts of Eastern Georgian Bay since 2013. We focus primarily on aquatic Phragmites growth, which allows us to effectively use manual cutting methods, including the cut to drown method in standing water and spading along shorelines when necessary. So since our work encompasses a lot of Georgian Bay, our efforts are tied to all the communities that are connected to the Bay. This makes our work necessarily cooperative. We engage with multiple municipalities, First Nations, community groups, and cottage associations that contribute greatly to the success of our Phragmites project. Our ultimate goal with each of our collaborators is to provide them with the education and the support that they need to lead the fight against Phragmites within their own communities. So we're constantly expanding in terms of where we operate. And as you can imagine, we dedicate a large portion of our time to keeping track of where Phragmites actually are. Our mapping efforts are typically performed from boats, which, depending on the location, can vary from being a literal breeze to semi-hazardous slog. One of these sloggier areas that we manage is called Matchedash Bay, and it's pictured in the top right of my slide. It's an environmentally significant Ramsar site containing diverse wetland, forest, and meadow habitats. It's one of those tricky sort of wetlands where if you go by foot, you're going to be thwarted by deep water, but if you take a canoe, you're going to be blocked by a wall of dense cattails. We knew this was a really important area to manage and to remove Phragmites from, but just locating the Phragmites took a substantial amount of time and effort. So eventually we learned about using drones for environmental monitoring from one of our collaborators, Nipissing First Nation, which Curtis talked about earlier, and we were inspired to do a similar thing to what they were doing. So we purchased a drone in 2023 and we got to it. So first off, we got our staff certified in drone operations. And right away, we found that we could easily map sites that we were previously unable to, or sites that required a significant amount of effort to reach. And we were able to map much larger areas much faster than we did by foot or by boat. For example, to capture imagery with a really high resolution, we can fly at 100 meters altitude with a high amount of overlap between our images, and a flight set with these conditions can cover 16.5 hectares or 40 acres in only 15 minutes, which is way faster than what we were mapping before. There are some limitations with using drones, however. The first one being weather. This includes lighting. So we need to fly with consistent lighting, either full sun or full cloud, to get good imagery. We also can't fly when there's moisture, so no rain, no snow, and we have to be mindful of freezing, so especially since the drone operates at higher altitudes. Our flight duration is also limited by our battery capacity and the number of batteries that we have. And we're limited by like finding appropriate launch sites. So this could be potentially challenging depending on where you are, but we haven't had too many issues with this since we're lucky that there's a lot of public lands around where we work. And two of the biggest restrictions on where you can fly include controlled airspaces in which you need special permission to enter, like over airports, over wildfires, and maintaining BLOS, which means keeping your drone within your visual line of sight. So again, neither of these things have been an issue for us where we work, but they're really important to be kept in mind when you're planning flight routes for your drone. And some things that we found work for us so far include conducting fieldwork in the fall for the best images. Curtis kind of talked about this earlier, and we'll go more into this later. And having 2 staff members out in the field operating the drone. So one person actually driving it and one person to make sure that we don't lose sight of it. In addition to launching the drone from the sides of trails or the sides of low traffic roads. And lastly, although we haven't had any close calls, we do operate in areas with floatplane traffic, and we're considering certifying our staff with aeronautical radio licenses so that we can communicate with any manned aircraft that could be nearby to further lower the risk of an accident. So if you're thinking that this sounds complicated or intimidating, it can seem that way at first, but drones quickly kind of show their worth as great tools that can be used safely with relative ease. So now I'll introduce the flight equipment that we use. So we use a DJI Matrice 300 drone with an RTK station. So they're both pictured at the top of my screen. This drone allows us to pre-develop automated flight missions and allows us to interchange payloads. So we can equip custom cameras or sensors that allow us to do different analyses. And the RTK station improves the GPS accuracy of our drone. I won't go into details on the specs of those, but we can follow up with any questions that you have after the presentation. And lastly is our sort of star of the show in terms of our presentation. It's the multispectral camera. So it's a MicaSense Red Edge MX with 5 different sensors, as you can see in the picture, and each record information from 5 different bands of light. That's red, green, blue, red edge, and near infrared. So these different sensors allow us to conduct the multispectral GIS analysis on the images that we collect that Jack's going to talk to you about.
Hi, everyone. My name is Jack, and I am the GIS Technician with Georgian Bay Forever, and I'm going to run through our image analysis classification process. But first, here's a bit of background physics. So radiation emitted by the sun comes in many forms. Some of this radiation can be seen as visible light, but the vast majority is not detectable by the human eye. Most surfaces, like vegetation, have a distinct pattern of reflectance across the electromagnetic spectrum. We call this a spectral signature. As we know, plants contain chlorophyll. Chlorophyll reflects higher amounts of green light than it does blue or red, giving vegetation its distinct color. What we don't see is vegetation's extremely high levels of scattering in the near-infrared portion of the spectrum. Cell structure is what governs this process. In the fall, when temperatures drop and sunlight is reduced, chlorophyll is lost and cells begin to break down, changing the spectral signature. We can use this process to our advantage when detecting Phragmites. Given how well Phragmites do in wetland environments, we can witness a prolonged green and near-infrared reflectance compared to neighboring vegetation. As Erin mentioned, the camera mounted aboard our drone is finely tuned to capture 5 spectral bands or wave frequencies that correspond to blue, green, red, red edge, and near-infrared light. Erin also mentioned, I think, that we have a 7-centimeter pixel resolution at about 100 meters of altitude. And our goal is to teach the computer how to classify each pixel based on its spectral signature. Specifically, we want to know which pixels are invasive Phragmites. Oh, there we go. During a flight, our drone will capture thousands of images, 1 for each band and hundreds to make up the study site. With Pix4D, we can seamlessly stitch all of these images together to form a mosaic. Pix4D will optimize our resolution and generate a digital surface model from a low density point cloud. In this image, you can see all the different positions our drone was in when the camera was fired, and the generated low density point cloud that forms our surface model. So once the image of our study site has been constructed, we can then begin to analyze its complexity. As you can see, there are many distinct features in our landscape: rock features, shallow water bodies with varying surface reflectance characteristics, individual Phragmites stocks, large homogeneous patches, and tree features, all with their own spectral signature. To train our model to classify all of these features in our image accurately, we need to provide thousands of accurately drawn training samples. This requires a trained eye and ground truthing, someone on the ground recording locations of features with a GPS. Once we know where and what everything is, a technician like myself can begin to digitize our training samples. So pixel-based deep learning requires a significant number of training samples. In this study site, we created over 3,000 samples from 4 sites and over 10 flights across Matchedash Bay. We do this so our deep learning model is robust and is prepared to encounter a variety of features when we try to classify new images. The model learns by studying the spectral signature patterns and elevation values of the samples from each class. The model also learns the physical structure of features and the spatial relationship between classes. By flipping and rotating our samples, we can generate even more unique observations. And from what started as 3,000 samples has now become 250,000 samples. Once our model has been trained, we can run it on one of our study sites. And depending on its performance, we can choose to fine-tune it, focusing on generating more samples in areas of weak performance. We can retrain and repeat this process until we are satisfied with the results. So after many separate attempts and band combinations, sampling techniques, hyperparameter adjustments, we were able to train and model with about a 93% accuracy, overall accuracy. This model was able to detect invasive Phragmites at 90%. As you can see, the top right image, I think, shows this well. You can see that, actually, I'll start with the bottom left. The bottom left, you can see that the model captures these large homogeneous patches quite well. And then the top right, smaller patches and individual stocks, tougher to capture, but we're still able to get the core of these areas. Other classes had similarly high performance, with the weakest performer being lilies and other floating vegetation. So why is this useful? First, having the data in this format allows us to quantify a large range of variables. We can measure total and individual patch area, parameter and shape variables, complexity, proximity, and many more. Second, we can track these changes over time. In ArcGIS, we can consistently extract the same area of interest from our study site, giving us an accurate year after year comparison. We can track growth rate, parameter and shape changes in vegetation health, and most importantly, we can track treatment progress. How much frag have we removed? How much is returned after a given amount of time? And how quickly can we see native succession? Third, we can use field observations to make more inferences. We can look at density and height to measure volume. We can look at the relationship between water depth and other variables. We can monitor changes in native wildlife behavior. The future research possibilities continue to expand as we explore new methods and learn from our progress. Yeah, thank you for giving us your time. And yeah, I think we're gonna open it up for some questions.
Sure are. Thank you so much. You were right on time. Good on you. That's really neat work. I'm so excited about this because, being able to really target Phragmites with the drones is going to be key to having that signature. There is a question actually from Chris from TRC. He was just wondering if you have any restrictions in flying around the hydro corridor. One of the photos showed the power lines there.
Erin, yeah, go ahead. I can speak to like a classification perspective, but yeah, go ahead.
Yeah, so I don't think there's any specific restrictions in like the drone rules about flying over the hydrocorridors, we just have to make sure that like our drone avoids them. So we program our drone to like, our drone flows, like we fly it automated, as I said before. And so we make sure that it's going, I forget how high we set it, but like well above the power lines so that there's no chance of it coming into contact with them. And it's really accurate and it's really good at staying in place. So we haven't had, like it's well out of the way of the hydro line. So there's no problem with that.
Great. Thank you so much.
Yeah. From a classification perspective, we actually have a lot of some imagery from above looking down on hydro lines. And it makes classification actually quite difficult, especially the digital elevation model is not really created accurately with those hydro lines. Yeah, so that is a challenge. We do have lots of sites, Phragmites, under hydro lines, which makes it difficult to map from the drone. From that
Fair enough. Yeah. Well, thank you both so much for coming today and presenting your really neat research that you're working on, and good luck with the rest of it. We're going to move on to the next session. I think there's another question there for you, if you don't mind. I just look in the Q&A section there to answer it offline, and we'll go to our next presenters, who are Dr. Michael McTavish and Dr. Ian Jones, and both with the University of Toronto, and both have been instrumental in rolling out the biocontrol research in Canada, in partner with a lot of folks in the US, and just delighted to have you both back again this year to give us an update on how things are going with biocontrol. I know there's so much interest.
Great. Thanks very much, Janice. I think, is that coming through all right for everyone?
Yes, there we are. We're good.
Yeah, okay, perfect. Okay, well, good to see everyone. We're happy to be joining this meeting again this year. We're going to be presenting about the biological control program for introduced Phragmites that our lab has been working on for a few years now. And today we're going to be looking at some of the supporting work and research that goes on to always iterate on and improve aspects of that this program. So as a quick reminder, for those not already familiar with it, biological control or biocontrol uses living organisms to help control a pest. And this is emerging as an important tool to help, an additional tool for what we already have to help manage introduced Phragmites here in Ontario. The goal here is to use the feeding damage from host-specific natural enemies to suppress Phragmites, so not fully remove from the landscape, but suppress it and its impacts. And some of the benefits to this tool are that it acts at a very large scale, it's long-term, and it is generally low cost. And because these are living organisms that go after one particular species, has very low non-target impact. So it's very specific as well. The biocontrol agents that we're working with here are 2 European stem-boring moth specialists. So they feed on only introduced Phragmites. We see here Arcanera neurica and Lanisa geminipuncta as caterpillars and as the adult moths. And as you can see in the middle, the larvae of both these species feed on the internal stem tissues of Phragmites. And so this will generally kill young stems, small stems of Phragmites outright, and then stunt the growth of larger, more robust stems, so leading to that population level suppression that we mentioned earlier. So since 2019, when we did the first releases, we've developed core operational protocols for how to rear, release, and monitor the insects. And we've used those tools to release approximately 30,000 insects across a network now of 50 sites. We're not talking today so much about the large-scale monitoring efforts on this so far, but we're seeing very encouraging results in terms of early evidence of establishment and impact. And you're going to see a little bit more of what that looks like at one of these sites in particular, from Claire Sean, so stay tuned for the presentation after this. But today we're focusing on one of the particular methods we use, which is egg releases. These are a very important strategy that we use for releasing the biocontrol agents. We can release all different life stages of the agent, but eggs are nice and convenient to use, and we can do mass releases very easily. So generally we're taking eggs and we're putting them out into the field, into Phragmites, in the spring before they hatch. We place them in these containers that have a mesh bottom on them. So as the larvae hatch, they'll crawl out and go and find a Phragmites stem in the field to start feeding on. And this works really very well, but we're always looking to improve the methods that we have as well. So just two of the questions that we've been working on to sort of further this work with eggs of the agents. I'm going to be looking at, can we use cold storage to manipulate the timing of when those eggs hatch? And then I'm going to be handing it off to my colleague Ian here to look at whether we can transfer stems with eggs on them from existing nurse sites to new locations as a viable release strategy. So eggs we often have from either our rearing or maybe that have been collected from a field site. Often we're overwintering them into the spring where they either go out before they hatch or we hatch them into larvae in the lab and then release those larvae in some fashion. But what we're looking to do is whether we have tools to control the exact timing of that hatch a little bit better. Being able to control that would help us synchronize larvae with the emergence of Phragmites stems in the field. So the larvae need certain sizes, ages, and characteristics of stems for that release to be particularly effective. And also the spring and the release time is very busy and we're working with large numbers of insects. So if we can control timing and stagger out those releases, that would be logistically very practical and useful as well. So to address this, we conducted an experiment that started in mid-March. We had eggs of both of the species. We had about 20 to 30 eggs in these dishes, and we assigned them to different storage conditions. So we either left them outside to experience natural outdoor temperatures, sort of the default what happens normally. We brought them indoors early in mid-March before we would expect them to normally hatch to see if we can speed that process up. But the main focus was really, can we slow them down? And so we used cold to do this. We put eggs into cold storage treatments for 7, 8, 9, 10, or 11 weeks before then bringing them back outdoors and allowing them to hatch. So throughout this process, we monitor temperature, we look at the timing of when eggs hatch, and how many of those eggs do finally hatch. So we saw when we don't do anything at all, the sort of natural hatch for both species tended to occur in early May, which is what we sort of normally expect. When we brought them in right away, we saw hatch within about 2 weeks of that time. So we're able to produce larvae just over a month earlier than we normally would. When we put them into the cold, we do see a delay in that hatch timing. At our longest period of cold storage, we saw delays of up to 31 days. So just about a month there. But we also want to know, does that cold storage have some sort of detrimental effect on total hatch? So we know for the two species, when they're outdoors naturally, we had hatch between about 65 and 95% between the two species. We saw for the eggs that we brought in indoors early, these actually experienced a large reduction in hatch. So you can get larvae early, but it's a little too early for them and you're going to lose a lot of that actual hatch. Fortunately, we saw eggs in cold storage didn't really experience a similar kind of reduction. There are a few anomalies that we think may be related to the cold storage treatments, but broadly, hatch was unaffected. So overall, we know we can bring in larvae early, but we're going to use a lot of them. That's okay because generally we want to slow things down rather than speed them up. And this can be done with cold storage up to a period of about a month without any negative impact on hatch. So that's a tool that we're going to be using going forwards to try and better line those larvae up with Phragmites emerging in the field and to stagger the logistical effort in the spring period. So I'm going to hand it off to Ian here now for a second question.
Perfect. Thanks, Michael. So one of the other kinds of egg releases that we're working on in the biological control program is actually transferring eggs from existing nurse sites into surrounding Phragmites infestations. So Michael showed you the kind of map of 50 release sites that we have across the province. And among those, we have some dotted around that are now kind of very established, robust populations of these agents. And we expect to see a natural dispersion of those agents across the landscape from those sites. But we'd also like to kind of actively promote that movement of the insects across the landscape. And the way we want to do that is by moving insects from nurse sites into surrounding Phragmites or nearby Phragmites infestations. So the question is, what's the best way to move insects from one site to another? So next slide, Michael. So these insects lay their eggs underneath the leaf sheaths of the Phragmites stems, and the eggs are the overwintering life stage. So it's underneath those leaf sheaths that the eggs remain throughout the winter until they hatch in the spring. So what we want to know is, can we collect stems in the fall, for example, in the hope that they contain eggs, and just move those into new sites and introduce the agents to the new sites that way? And to first kind of to get to the bottom of that, we wanted to ask two main questions first, which were how effectively can we harvest eggs from collected stems? And also, if we're able to harvest those stems, whereabouts on the stems are the eggs most commonly laid? So the stems you can see on this slide are collected from a nurse site in Waterloo. So there's 100 stems there, which we took and dissected carefully. And within those stems, we found 316 eggs. So we're really, really pleased and encouraged by the rate at which we were able to harvest these agent eggs from collected stems from nurse sites. What the graph on this slide shows is the positions on those stems where the eggs were found. And you can see the vast majority are found quite low down on the stems, certainly within the lowest meter. So what that means is if we want to transfer stems from one site to another, we don't have to transport the whole 4-meter tall stem. We can focus on harvesting that lowest meter, and we're going to be encompassing the vast majority of the eggs that way. Next slide. So last summer, 2024, for the first time, we actually tested this process of transferring stems from nurse sites into new infestations. So this was done at Ontario Tech University. So these kind of planters you can see here are on the Ontario Tech campus, and they're completely inundated with invasive Phragmites and have been for years. We mowed that Phragmites in the winter, and then we transferred in 18 blocks of 75 stems that we collected from nurse sites. So those stem transfers were transferred in 3 different treatments based on when the stems were collected and where the stems containing eggs were overwintered. So the first treatment, the stems were collected in the fall, shortly after the eggs would have been laid, and then just moved directly into the Ontario Tech site. So those eggs would have overwintered at Ontario Tech. The second treatment, the stems were collected in the fall again, but they were stored at the University of Toronto in sheds before being moved into the experimental site in the spring. And then the final treatment, the stems were collected in the spring, shortly before the growth season, and then just moved immediately into the Ontario Tech release site. Next slide. So as the Phragmites grew up in those planters, as you can see in this slide, we monitored each of those stem transfer blocks for signs of damage from agents that had been transported along with those stems. So what we're looking for are these really kind of characteristic boreholes that you can see on the right-hand side here. Really encouragingly, we found lots of damage in all 18 of these stem transfer blocks. So that shows us really nicely that this is going to be an effective way of moving insects from one site to another. And what we also found was that those 3 treatments that I described all worked equally well. There were no significant differences among those. So what this does for us is it tells us that it keeps open a whole load of opportunities in terms of when we can collect the stems from nurse sites, where we can overwinter them, and when the transfers take place. All of these things seem to be effective ways of moving the insects. So next slide, Michael. So the final thing we did was we checked and we monitored all of our stem transfer release plots at the end of the growth season for impacts on the Phragmites itself, compared to control plots within this same patch of Phragmites. And what we found was where we transferred those stems and where we had the damage from the biological control agents, we saw significant reductions in stem density and panicle formation in those plots. Now, this is kind of really preliminary impact data in that it's happening in the year of the release, and it's only at the level of the plot. But it's really encouraging to see the insects having this kind of effect on the plant. And this, I think, is a kind of a nice gateway, this kind of very preliminary work, into some of the really robust impact work and data that you're going to see from Claire and Waterloo in a few minutes, I think. So next slide, Michael. Yeah, so just to summarize, the work with the cold storage work has showed us that we can delay the egg hatch of these agents for as much as a month or more without reducing egg viability. And what that does in the spring is it kind of expands that window during which we can conduct both egg releases and larval releases, which is really huge for the program in terms of kind of how efficient we can be. We also know now that we can harvest eggs by collecting stems from nurse sites, and those eggs can be used to release in the traditional way, or we can do these stem transfers. We know that we've identified this effective method of moving insects or moving eggs from one site to another. Next slide. Yeah, so I know we don't have too much time left. I just want to say a big thank you to all of our collaborators, particularly people like Ducks Unlimited Canada and NCC, who've been fantastic partners to us for many years and have helped us find sites to release in and been working very closely with us throughout that time. I can't mention everyone, but I'd be very happy to take any questions that you guys have.
Thank you so much, Michael and Ian. That's really exciting work that you're doing. And again, really gaining every year more and more information and knowledge is going to be so useful. There's a question, Nicole Angel, she asked, do you have any indication of the potential for release within the United States?
Yeah, so the kind of the insects were approved by the tag agency there, but they still have an extra step to go through in order to be approved. And so there's one of the differences in the kind of approval system between Canada and the US is that the US system doesn't necessarily consider the effects of not releasing the insects in the way that the Canadian system is kind of permitted to do. But we're still hopeful that we'll get the insects released in the US at some point. We're working at the moment to generate some extra host range results that will kind of appease some of the final queries that they have.
Right. That's a nice segue to the second question, which I don't have time to ask, but if you don't mind, actually, there's quite a few of them now coming up. If you both don't mind, just go in the Q&A and answer.
Oh, yeah.
Thank you so much. And
Okay.
I'll move us on to the next session, which is Claire Sean, which mentioned that she's also partaking in a lot of the great research that's going on. And Claire is a PhD candidate at the University of Waterloo and Dr. Rebecca Rooney's lab. And welcome, Claire, and thank you for updating us on your research as well.
Hi, can everyone see my screen? Hopefully you can see
Yes.
it. Perfect. So yeah, I'm going to be continuing on the research updates for the biological control program and also kicking off the University of Waterloo research updates. And so we just heard a lot from Michael and Ian about the egg part of the life cycle. But for my presentation, I'm going to be thinking mostly about the larva part of the life cycle for the biological control agents. And that's because as larvae, they're feeding on the inner stem tissue of the Phragmites plant or the Phragmites stems, and ultimately they're damaging the vasculature of the Phragmites plant. And in the field, we tend to see one of three responses of the Phragmites stem to this larval feeding. The first one is that the stem dies following larval feeding. Next would be that the stem forms secondary branches or side shoots following larval feeding. And another response we see is when the top half of the stem wilts and the lower half remains alive. We know from Europe that this reduces the height, the above ground biomass, and the flowering or the seed production capabilities of the Phragmites stems. And in a biological control program, we would hope and expect that this would reduce the competitive ability of invasive Phragmites, ultimately allowing for plant community recovery to occur. And so this year is kind of the overarching objective for my PhD work, where I'm trying to characterize the efficacy of Phragmites biocontrol. And so today I'm going to be sharing a kind of quick update on that progress to date and an interim update as this work is still continuing. For the sake of a little bit of focus, I'm focusing on one site that I've conducted research at for the past 3 years. It's called Rare Charitable Research Reserve. It's a private land trust in Southern Ontario, located in the Cambridge area of Southern Ontario in the Waterloo region. And on this property, I actually have two field sites, the wet site, which is a Phragmites patch, part of a larger wetland complex, and then a dry site, another Phragmites patch located at the bottom of a hill slope. And these sites were set up in 2022 in the same way, where we did these egg releases. Maybe you might recognize those egg cups Michael just talked about. And we had 5 egg releases of Lineasa geminipuncta at each of those sites, and then a paired number of control plots. And we used 1 by 1 meter squared quadrats to monitor the health and abundance of the invasive Phragmites after we did these biocontrol releases. So I just mentioned we released the biocontrol agents in these patches in 2022, and then we didn't do any supplemental releases after that. So by 2023, if there was damage in the patch, that was the moths continuing their life cycle on their own. And by 2024, that would have been the second field generation of the moths. And so we repeatedly sampled these quadrats that we used to characterize the health and the abundance of the invasive Phragmites over the past 3 years. And the first thing I can say, one of the most positive updates is that the moths have persisted over 2 field generations, which has been really good to see. And we've observed damaged stems within both of these patches in both 2023 and 2024. You can see in the photo that's an indicator that a moth has fed on that stem through those larval boreholes. As well as actually seeing the physical larvae themselves with this little photo shoot I did of one of them. And not only have we seen damaged stems, but this damage has spread throughout the patches of Phragmites and increased in the 2 years following this initial release. And so we've seen over time that the density of damaged stems has increased at both of the sites. And so in both 2023 and 2024, we had damage at these sites, like I mentioned. And at the dry site in 2024, we had about 56% of stems damaged. So more than half of the stems in our plots were actually damaged by the biocontrol agents. And at the wet site, it was about 82%. So over 4/5 of the stems in our plots were damaged by the biocontrol agents. And so one thing we've been recording as well, besides just are these stems damaged or not by the biocontrol agents, but what proportion of these stems are branching versus dying versus this wilt option. And so we've seen that the proportion of stems branching have increased over time. If you look at the dry site, it was initially very low with a low proportion of our damaged stems producing those secondary branches, but it's kind of increased since then. Same with the wet site as well. We're seeing increases where now even at the wet site, over 80% of our damaged stems are producing those secondary branches. And that's a bit of a trade-off that we've seen at this site in particular, because that means less and less stems are dying each year as a result of that larval feeding. And so while they certainly still are impacted by the larval feeding, we're seeing the increased production of secondary branches and less of this dying following larval feeding. One really encouraging thing that we've seen is that in our high outbreak years in 2024, we had really strong limitations to the flowering density or the seed production capability of Phragmites stems. And so you can see on the right side there, the panicle or the Phragmites seed head. And in 2023 at the dry site, about 25% of stems were producing a panicle. And by 2024, that was only 3% of stems. At the wet site, it dropped from about 15% in 2023 to just over 1% in 2024. So really strong reductions to the amount of seed that Phragmites is able to produce, and ultimately the amount of seed that might be able to spread to nearby patches, which is what we want to avoid. Another thing that we've seen that's been really encouraging is that between 2023 and 2024, we had really strong reductions in our canopy height. So this is a measure of where the canopy, the live Phragmites canopy is kind of sitting. And at the dry site, we had about a 30 centimeter reduction in the canopy height. And at the wet site, that was even more. We had almost a meter, about 96 centimeters reduction in canopy height between 2023 and 2024. And we didn't take this measure in 2022, unfortunately. But we have been calculating the average stem height of the stems in our plots. And we're seeing a similar trend where, again, the canopy height and the average stem height is shortened by the biocontrol feeding. And for this case, a picture kind of tells 1,000 words here. These are 2 photos I took, one at the end of August in 2022 and one at the beginning of September in 2024, right after I pulled up to the site to actually sample it for this year or for the previous year. And so you can see in 2022, it was this very healthy, very lush green stand of Phragmites with a lot of panicles being formed. And by 2024, that has really changed. There's this kind of layer that we see above the Phragmites with these standing dead stems, which are stems from previous years. And the canopy height is a lot shorter. We're seeing a lot less production of those panicles, like I mentioned. And then you can see that those stems are towering about a meter over, which is, what our data says, but you can see it as well. Those are the standing dead stems from previous years that would have been healthy stems in previous years. So that's been a really encouraging change, one that you can see if you're just driving past that patch of Phragmites. And so another thing that we're interested in is how much light is the Phragmites canopy blocking, and is biocontrol allowing for more light to reach the ground? And initially, which maybe is not a surprise to many people who have worked in Phragmites, Phragmites blocks a lot of light. In our plots in 2022 and 2023, we found that over 99% of light was being blocked and not reaching the ground. And we're starting to see reductions to that, which means that more light is actually reaching the ground, more light is penetrating throughout the Phragmites canopy. And so these are small reductions. Now we're sitting at 95% of light is blocked and 94% of light is blocked. So about 4 to 5% more light is starting to reach the ground with the thinning of the canopy. And so that is, a small change, but something that we hope native plants can start to take advantage of, and something that we're going to be really interested to monitor in future years. One thing that we haven't seen at this site yet really in any capacity is any change in the stem density. So this is a measure of the stems produced per meter squared. And so really it's a reflection of the previous year's investment into the rhizome and how much the Phragmites clone is sending up. Tells us how healthy really the stem was in the past, the stand was in the past few years. And we haven't seen any responses yet. We haven't seen any marked differences in stem density, but that's something that we might actually see a kind of an effect of the 2024 outbreak in our 2025 field season. So again, something to monitor, but we haven't seen any changes yet. This is also looking at live stems per meter squared. So we've kind of subtracted any stems that died following that larval feeding or died for any other reason like trampling. And we haven't seen really any changes in this metric either. And so with that, I wanted to end with a quick summary. We have seen that a high proportion of stems are damaged just after 2 field generations of the moth, and we're seeing larger and larger populations every year through the increased damage density of the Phragmites stems. We've seen really strong reductions. It diminishes to the sexual reproductive output of Phragmites with this high biocontrol damage. So hopefully it means there's less seed entering into our wetlands nearby and ultimately starting new Phragmites patches. And we saw really strong reductions to the canopy height and mean stem height with biocontrol, which ultimately allowed for some increased light penetration into the ground. One thing that we haven't seen is that stem density really changes. And this is just a quick reminder is that with biological control, eradication is not the goal, nor is it really likely to occur. And so that kind of reinforces that. And then in terms of next steps, maybe what I'll be presenting at in OPWG next year, we're going to continue to monitor the Phragmites health and abundance at this site that I highlighted, as well as 10 more sites in Southern Ontario. We expanded this past field season. And then continuing to monitor, I didn't present it, but we have also been collecting plant community data about our diversity and abundance of native plants. And so we want to continue to monitor plant community changes and native plant recovery over time. And so with that, I just want to thank our research partners, collaborators, and volunteers who helped out with this work, as well as OPWG for allowing me to provide an update.
Thank you so much, Claire. That's great research. I know the site that my sister lives down the road. I pass it quite a bit. It's pretty neat.
Good to hear.
Yeah, I'm really interested to see, the next research where with the shorter stature and maybe a little bit more light coming through if the native plants are able to kind of eke out in existence as well. That's really the ultimate goal, to stop this plant from being a bully. If we can achieve that, the biocontrols, that'd be a huge advantage for sure. So thank you. I don't see any questions specifically for your research, but I am quite sure there's going to be some coming. So if you don't mind, just to keep an eye on the chat section or sorry, the Q&A section, that'd be great. So thank you so much. Keep up the good work. We appreciate it. Thanks for attending our meeting today and giving us an update. I see next we have a lab mate of yours, Andrei Marenko. Sorry if I don't pronounce your name correctly, Andrei.
Thank you so much.
Welcome to our meeting, and thank you as well for your research and coming to talk to us about what you're finding, I guess, in the Long Point area, the recovery of the native species, and and how important they are for the waterfowl there. So I'll let you get away. Thank you so much.
Thank you so much. Can you see my presentation?
Yes.
Thank you. Hello, dear colleagues. I appreciate the opportunity to present my research project here today, titled The Assessment Tools for Evaluating Recovery of Wetland Forage Value for Waterfowl Following Invasive Species Suppression. So, more than 30 species of waterfowl live in Canada, and the wetland ecosystem of the Great Lakes region and Lake Erie in particular are known for their diversity and richness of natural resources. These create the necessary conditions for nesting, migration, and feeding. The hydrologic properties of Lake Erie coastal zone provide the existence of the diverse wetland ecosystem with the different landscapes and plant communities that definitely have a high food value for water birds. At the same moment, the Long Point and Big Creek National Wildlife Areas are essential wetland habitats in southern Ontario that serve as stopover sites for at least 17 species of waterfowl. However, these conditions support the spread of invasive plants such European Phragmites australis, which endanger this waterfowl population. The combination of the vegetative and sexual reproduction mechanism, high intensity of biomass accumulation, the strong capacity to tolerate stress, and a wide range of Canada's climatic conditions allow Phragmites to outcompete native plant species, potentially depriving waterfowl of food. How my colleagues already mentioned today, there are different methods to control the spread of invasive Phragmites, and the most common way today is the herbicide treatment. However, the native plant species don't necessarily recolonize. Instead, the suppression of Phragmites may lead to spread the other invasive plant species in ecosystems, such as European frogbeet, which you can see on the right corner on the slide. And these secondary invaders may become dominant, and they also have low nutrition value for waterfowl. The Vegetative Forage Quality Index, or VFQI, was developed in the USA to estimate the food value of plant communities for waterfowl. This index is based on the calculation of the modified feed quality coefficient C, derived from the expert opinions for each plant species, with values ranging from 1 to 4 for each plant species, where 1 represents poor forage value and 4 represents excellent forage value. So we use this index to monitor waterfowl forage quality during 2022 and 2023 in 3 vegetation types: Phragmites australis invaded, herbicides treated, and reference sites with natural vegetation. In total, we have 48 50 meters transect established in the Long Point and Big Creek National Wildlife Areas in 2022. On each transect, plant biodiversity data were collected using point intercept method along each transect twice. First time in August 2022 and second time in August 2023. So at the beginning of the experiment, we had 16 transects of each vegetation type, including experimental controls in areas where the managers left Phragmites intact. But to meet their wetland managers' conservation goals, half of the experimental controls from the first year were actually treated in the second year, creating a shingle design. So to test if waterfowl forage quality index scores differed among the reference, control, and herbicide-treated vegetation types, we first calculated the VFQI scores. However, we encountered a hurdle because many of the plant species we found in our transects didn't have published C coefficients. In fact, only about 50% of the observed plant species had a published C coefficients for the VFQI calculation. So we had to leave a lot of species in the calculating the VFQI values. And importantly, most of the species that did have coefficient were invasive plants, such as Phragmites and frogbeets, which were highly abundant. But of course, there was a lot less Phragmites in the treated and referent transects, introducing a bit of bias into our VFQI calculation. And this doesn't allow us to fully assess the nutritional value of plant communities as food for waterfowl. So we analyzed the resulting of the VFQI scores with a 2-factor analysis of variance, where the main factors were a year 2022 versus 2023 and vegetation type, treated, control, and reference. A significant interaction would mean that the effect of vegetation type changed through time. And we expected that the VFQI values would differ among the vegetation types significantly because we expected the reference transect to have higher VFQI values than the invaded control transect, and we expected the treatments to improve the VFQI values through time. The forage quality index values were variable between the years and among the vegetation types, but we didn't really detect a significant interaction between the years and vegetation type. So indeed, the full linear model wasn't supported by data and isn't statistically significant, suggesting that herbicide application to suppress invasive Phragmites didn't enhance forage quality. But because so many of the plants were missing coefficients, we were not confident that this interpretation was accurate. So we decided to develop the coefficients for the most widespread and abundant wetland plant species in the region to fill in the gaps and recalculate the VFQI. So to determine the most common wetland plant species for Southern Ontario, we requested data from the Great Lakes Coastal Wetland Monitoring Program. Based on the most recent observations from 46 coastal wetlands in the ecoregion 7E of Ontario, we identified 153 plant species for the emergent vegetation zone and 65 plant species for the submergent vegetation zone. After the analysis of all these plant species, along with 44 additional species lacking coefficients that we observed during our fieldwork, we obtained a list of the 74 the most common and abundant plant species for the region. So we use the same expert elicitation approach that was used to initially develop the VFQI tool by Fleming and Farley. We engaged experts recommended by the governmental and non-governmental organizations related to the waterfall conservation and habitat preservation, as well as authors of articles that have been involved in developing the assessment of the nutritional value of wetland plants as food for waterfall. A total of 22 experts participated, each holding a master's or PhD degree or having over 5 years of experience in the field. In the survey, experts were asked to assess the forage value of each plant species on a scale from 1 to 4, and to determine the final coefficients for each plant species, we used the median score. So after determining the coefficients and conducting recalculation, we rerun the two-factor ANOVA with interaction and found no change in the interpretation of the results. If anything, the model fit was worse than before. So we made a step back, and on the reflection, we thought there might actually be a problem with the formula used to calculate the VFQI, and I will try to illustrate it with the data from the 3 real transects. So the first one is the control, the middle one is the treated transect, and the last one, this is the reference transect with the natural vegetation. So the control transect consists of the monoculture of Phragmites with a forage coefficient 1. The VFQI calculation is also equal to 1. The treated transect after the use of herbicide was occupied by 3 plant species with the coefficient 1, 2, and 2. So intuitively, this should be better than a transect with 1 species with a C coefficient value of 1. But because of the sum of the weighted proportional abundance of each plant is divided by the total number of the plant species on the transect, which is for control transect only 1 because we have Phragmites, and for treated transect it's 3 because we have 3 plant species. The VFQI calculation for this transect, the treated one, is actually lower and equal 0.69. At the same moment, the reference transect usually consists of the plant species with the higher coefficients, but at the same time, biodiversity is also higher and ranges from 10 to 20 plant species per transect. So the sum of the weighted proportional abundance is divided by 10 to 20, and the result is an even smaller VFQI score. So in an analogous tool called the Floristic Quality Index, researchers had proposed a different calculation that doesn't divide by the total richness of the transect. This alternative measure is called the weighted mean coefficient of conservatism, and it's noted as the less sensitive to differences in the plant species richness. So we decided to adopt this formula to represent relative forage value. So we repeated the two-factor ANOVA using weighted mean coefficient of conservatism values as the response variable, and this time the results better aligned with our observations in the field. The full linear model was statistically significant, as were each term in the model, and the control transect had lower forage value than the reference transect and remained consistent year to year. The treated transects improve with time, getting closer to the reference transects for each value within 2 years of treatment. And this makes in general, more intuitive sense, given the data. So we are recommending the VFQI be replaced with the weighted mean coefficient of conservatism index to represent the forage value of plant community for waterfall. But at the same moment, regardless of which formula is used, these coefficients are still all based on the expert opinion, and we wanted to validate this a bit by actually measuring how much seeds and tuber materials is in the sediment. So in September 2022 and 2023, 3 superficial soil samples were collected from each transect, one at the beginning, one at the middle, and one at the end of each transect. In total, we collected 144 samples each year. So my next goal in this study are to compare the weighted mean coefficient of conservative scorms with the dry mass of the edible fraction of the sediment cores, and to investigate their relationship between vegetation types and location. I also aim to compare the coefficients obtained from the experts with the other available indexes that allow to identify the nutritional value of plants, such as true metabolism energy index, which reflects a real amount of energy which specific species of the waterfall can get from the specific species of plant. As well as compare the coefficients with functional traits of plants, such as number of seeds, number of tubers, amount of proteins in the seeds, etc. Thank you for your attention. I also would like to say thank you for all my colleagues who support my research, and to the partners. And now I would like to answer your questions.
Thank you so much, Andrew. Well, that was really fascinating. I do vegetation monitoring as well. I use the first quality indexes, and I was so perplexed for the same reasons. Like, I saw improvements in the diversity, but not so much in the number increasing. So thank you for doing this. It's so important, and taking it to the next step. So it's going to have so much value for us moving forward to try to use these values to show improvement. We're seeing with our eyes, but it's so important with these calculations so that we're consistent across the board in how we're representing these ecosystems and comparing. So that is really, really cool stuff. I'm really looking forward to seeing the results of your samples with what comes out of those sediments that you've collected as well. Thank you for that. It's neat that you're all in the same lab. Like Jersey's coming up next, also looking at the recovery of these ecosystems, and so it's really cool that you have each other to bounce ideas off and to work together. And that map that you showed, how it moved around, I'm really impressed with that. It's kind of cool stuff. Thank you so much.
Thank you.
I don't see any questions right now. I'm sure there will be some coming up. So if you don't mind, just keep an eye in the Q&A box there. Our next presenter is also from Dr. Rebecca Rooney's lab, Jersey Allison Fonts, and I had the great pleasure to meet Jersey at one of the research projects she was doing. Actually, we used the drone to spread some seeds. It was just kind of cool. But anyway, welcome, Jersey. Looking forward to hearing all the great research that you've been undertaking as well.
Awesome. Thank you so much, Janice. And no spoilers. You'll see at the end of the slides what's coming up. But just kind of in general, I'll be wrapping up the results of our long-term monitoring project, as well as some new things to come. So as we all know, Phragmites invasion has been extremely detrimental to biodiversity here in Ontario. And so the Long Point and Rondeau Provincial Park regions of Southern Ontario have been targeted as areas most needing treatment. And so the Long Point Peninsula, which you can see here, located along the shores of Lake Erie, is this diverse area known for its rich ecosystems such as sand dunes, forests, and of course, wetlands. And it is recognized as a wetland of international importance. And similarly, Rondeau Provincial Park holds significant ecological value as a provincially significant wetland, as well as an important bird area for congregatory species. And so both of these regions are home to some of Ontario's species that are either of special concern, threatened, or endangered, many of which have been negatively affected by the invasion and spread of Phragmites. And in 2016, a pilot project was launched to control Phragmites in these areas using an integrated approach that involved spraying targeted Phragmites areas with a glyphosate-based herbicide, followed by the rolling or mowing of these dead stems in order to encourage the return of native plant species. And so the plant community here regenerated what we say passively based on factors such as the local seed bank, surrounding plant populations, as well as changes in water depth. And so to assess the effectiveness of this approach, our lab initiated a long-term monitoring project, which tracked the recovery of the vegetation community in these 1 meter square plots, which you see on the right here, comparing treated areas with untreated control plots. And so a total of 80 treated plots and 40 control plots have been monitored annually since 2016. And so after 9 years of long-term efficacy monitoring, we have found 3 key findings. So firstly, after what you can see here as years of Phragmites invasion, pictured in yellow, we've observed a significant reduction in Phragmites density. So for example, 1 year after treatment, Phragmites stem density in these treated plots dropped by 99.7%. And so by the 2nd year, some plots did see a reappearance of Phragmites, but overall the plant was largely kept in check. Secondly, although Phragmites was effectively suppressed, treated areas then began to be dominated by other non-native species depicted here in orange, creating this secondary invasion. But fortunately, over time, the vegetation in most of these treated plots shifted again, which you can particularly see at this spot, with native species in green gradually establishing dominance. And so some of these native species included pickerel weed, various floating pond weeds, and wild rice. And so as our long-term monitoring project progressed, we found it necessary to reassess our objectives and to determine whether it was still relevant to continue evaluating Phragmites suppression efficacy. And so it became clear to us that Phragmites had been effectively controlled in our plots and that the plant community had recovered, regardless of what was in that community. So moving forward, our focus shifted to understanding whether the post-treatment vegetation community was now then aligning with what we call a reference community or developing into something entirely new. And so by reference community, what I mean is an area of vegetation that where Phragmites had never invaded. And so the new objective of our long-term monitoring project was to determine whether the vegetation in our treated areas was then aligning with these reference conditions or somehow evolving into a novel plant community. And so to assess this, we compared the plant communities in our established treated plots with those in new reference plots. And so in the summer of 2023, an example of one you can see on my slide, we set up 40 reference plots by matching them with the water depths of the treated plots we had assessed previously. And so happily or luckily, we found that there was no significant difference of our diversity indices or our richness indices between these treated and reference communities. And so this tells us that diversity as well as richness is on par with these uninvaded conditions, alluding to the treated community converging with our reference community. We also found that there was no significant difference of weighted mean coefficient of conservatism, or WMCCs, between our treated and reference communities. And so this is a variation of the floristic quality index, which generally is a scale that tells us how successful our restoration project is. So in general, or very plainly, the greater the value, the more successful your restoration outcome is. So in this metric, plants are given a coefficient of conservatism based on expert opinions that speaks to how sensitive that plant is, and therefore that plant community is to disturbance. And as you can see in our graph, we have a pretty wide range of WMCCs, which is reflective of a healthy and diverse ecosystem. And so finally, we measured vegetative foraging quality index, also known as VFQI. And so this is a relatively new to our study area measure that my lab mate Andrew was previously talking about. And similar to our other measures, there was no significant difference between reference and treated communities. However, we do take this result with a grain of salt, as the process of calculating VFQI for Long Point and Rondo is still pretty much in the works. And so as this index is further developed, it is a hope that the VFQI will more accurately represent what is in our vegetation communities. In general, though, it seems like the vegetation community of these treated sites is following a similar trajectory to what we see in the reference conditions, which is really awesome. And so after learning so much from our long-term monitoring, what does this mean to all of us in the grand scheme of things? Well, we've learned in our study system that firstly, effective plant community recovery needs more than just invasive species removal, because we can see that there are possible secondary invasions that were common for these herbicide-treated areas, maybe influenced by a lack of active revegetation, but I'll touch on that later. And finally, the vegetation communities in our treated areas were not significantly different from vegetation communities where Phragmites had never invaded. And so I have painted a very optimistic picture here that after these initial secondary invasions, because treatment was so successful, that the native plant community can begin to recover 3 to 5 years post-treatment, and then that's it. And without considering the broader context, this might seem like a highly successful approach that could be applied universally to all areas impacted by Phragmites. But as we all know, for those of us who are working and managing Phragmites, our results are extremely context dependent. And so this work was carried out in relatively pristine conditions, a largely undisturbed provincial park that serves as a biodiversity hotspot can pretty much be considered our best case scenario conditions, leading to a best case scenario conclusion. And so in areas lacking these ideal conditions, such as those affected by agricultural runoff or created wetlands with limited seed banks, such as those adjacent to nuclear stations, this passive approach to revegetate these areas after treatment may not be enough. And so in these cases, we may need to explore alternative and more active revegetation approaches. And so speaking of these created wetlands, ideally we would want to skip that secondary invasion stage and transition more immediately to native plant dominance, especially if we don't always have the resources for long-term monitoring. So one of the ways that we might be able to do that is through seeding these treated areas with a curated mix of native species. So the seed mix, for example, that we're using in our created wetland has species that have traits that we hypothesize will contribute to biotic resistance against non-native species such as Phragmites, but also to other ones. And we were given the opportunity to contribute to the IPCC's and Ontario Power Generation's restoration of this previously Phragmites-dominated wetland, which you can see on my slides. So in 2022 and 2023, the IPCC treated the Phragmites surrounding Coots Pond, a created wetland associated with the Darlington area of Ontario Power Generation. And so since the water cycle dynamics in the stormwater pond are different than a coastal wetland where the rest of our work takes place, we wanted to see if there would be a difference in seeding methods. So for example, broadcast seeding is a standard in restoration work, but another method that is of special interest to those we're working with is drone seeding. And so in a larger setting, if drone seeding is just as effective as these conventional methods, the work could potentially go much faster, and areas that are generally inaccessible can become accessible to us. And so at Coots Pond, we set up 12 plots, 4 that have been drone seeded, 4 that have been broadcast seeded, and 4 are controls, which we did not add any seeds. And so late autumn of 2023, I, along with Janice and Brian from IPCC, returned to Coots Pond to seed my plots. And so on the left is a picture of the drone that was used to spread my seed mix across 4 plots. And on the right is a picture of me broadcast seeding another 4 of my plots. And so last year, our lab returned to the plots to monitor the returning vegetation. And so similar to our work in Long Point, the first year after treatment didn't result in a very diverse plant community. And so we found that there wasn't much of a difference in our diversity or richness indices when comparing either the hand broadcast seeded or drone seeded plots to the plots without added seed. And we also found that there wasn't much of a difference in our floristic quality or foraging quality indices either. However, as optimistic as we are hoping that with time, the seeds that did germinate and prosper to the flowering and then seeding stage, as well as those more slow growing perennials that we added, will grow this year and contribute more to the overarching plant community as we return this summer to Coots Pond to do more vegetation assessments. And so with that, I would like to say a quick thank you to everyone who has contributed to this project, whether that was in terms of funds or fieldwork help or just general support. And to thank you all for listening to me today.
Thank you so much, Jersey. That was really interesting. I was at Coots. I saw a really nice regrowth of the vegetation. So it's just so interesting, the monitoring piece. But I agree with you. I think a little bit more time and it's going to look a lot different. There are a couple of, well, actually, there's quite a few questions.
Okay.
So I was so enthralled with listening to you. I haven't been keeping up here.
No, I know, it's okay.
Let's see here. Yeah, there's quite a few. And I don't see the secondary invasion naturally diminish or retreat.
Yeah, so there wasn't additional like follow-up in terms of like adding seeds or planting plugs or anything. In some of the areas there was additional treatment, but it was mostly like spot treatments. It wasn't as large scale, especially in terms of like the areas we were looking at.
The herbicide that was used prior to the vegetation the plant reseeding. Do you want me to answer that, or do you have that one?
Oh, which one it was?
Yeah, was it glyphosate or imazapyr?
Yeah, it was glyphosate.
Are your seed mixes available? And what's in your seed mixes? Would you mind, like in the because I think that might be of great interest to a lot of people, if you could answer that in the Q&A section?
Yeah, sure. I can type about that, but I can also answer it a little bit. So our seed mix is right now still super experimental, but it's based on Phragmites research coming out of Quebec, which during the mid to late 2010s, and is based on having like a diversity of functional groups and functional traits. So from the top of my head, some of the seeds that I added into the mix that also came up at Coots Pond was one of our ranunculus species, Persicaria species, and our mimulus species as well.
Nice. Well, those are three good plants to come up for sure. I think there's quite a few other questions, if you don't mind, just to go in there and answer those while your colleague from your lab is coming up next. Grace, thank you so much, Jersey. Really interesting research and very important research as well.
Thanks, Janet.
Thank you. So last but certainly not least is Grace Cole, and I actually also got to work with Grace. I mentioned earlier that she was quite involved with collecting the research for the spray drone work, and she got her master's off of that research, and I now understand is doing a lot of other neat work in Dr. Rebecca Rooney's lab as a research associate, and is here to present on some new findings that you have from a lot of the work that's going on in that lab. So welcome, Grace.
Thank you very much. I'm going to get my screen shared here. It's really nice to be back at OPWG, and I'm OPWG, and I'm really excited to showcase some of the work that I've been doing in the Waterloo Wetland Lab. I'll go through this pretty quickly. I have about 5 minutes to overview. But essentially, in collaboration with Canadian Wildlife Service, we've been working on creating a story map to kind of highlight a lot of the positive management outcomes that have come from the frag treatment in the Long Point region, and kind of showing it on a nice geospatial scale with some different visualizations of the changes in the vegetation and the different species in that area over time as a result of the treatments. So I'm not going to read through everything here, but we do kind of get into kind of the different focus, different maps here, so showing Ontario's priority place, the different national areas over here as well, and then parks that are priorities too. I hope it's not too laggy on the screen here. I'll try and scroll a bit slowly, but lots of information here about the different threats to biodiversity that FRAG poses and all the different efforts that have been introduced over a different timeline and strategies that have been in place to try and improve the overall biodiversity and conserve the area of the wetlands that we have in the Long Point area. Of course, this audience is very familiar with all of the negative impacts that FRAG has on biodiversity. And here we're kind of just highlighting the integration of best management practices that started back in 2016 by NCC. We have a really telling photo, I think, of the Long Point area and just how extensive the invasion of FRAG was. So that was back in 2019. Something we've tried to integrate a lot into the story map are these interactive elements. So here we have a slider of one of the monitoring plots where our first image is a plot in 2019. And then the users can scroll over and see what that same exact area looked like 4 years later in 2023. Next, we have a large map showcasing kind of the total classified area of Phragmites that we had in the region. And I hope it will play nicely on the screen for you all here. But here we're showing all the area that ended up being treated with herbicide, and some graphs here showing the total area in hectares. So you can see it was a really massive effort that's happened from 2016 to 2024, along with some different navigational units. So if you wanted to kind of zoom in just the Big Creek area and get a more detailed view of that, that can also be done in here. Of course, these efforts would not have been nearly as successful without all of the collaboration that took place. So that's something we really wanted to highlight as well in the story map, is just how many different organizations and groups were involved in this work. And that's really what we can attribute a lot of the success to. The last section here is really the positive management outcomes. So this was done with a lot of the different monitoring that's happened in the area and different research that's been going on in Long Point over the years. So as Jersey was just talking about, we have some information on the suppression efficacy of the treatments and some nice, that nice graph that she showed as well. Here we have an animation of the species dominance over time. So as she was mentioning, we're kind of seeing that transition from FRAG dominated to invasion dominated to more native dominated plots as time goes on after the treatments in these areas. Of course, we have some additional resources. If people are interested in learning more, then the information that's also available. We have information about the forage quality for waterfowl that Andrew presented on. Here we have just a visual of all the different transects in relation to FRAG we had in the area. Now we'll scroll through this. And again, we can see some of these graphs showing how we're slowly seeing the treated areas creep back towards reference conditions, which is very, very promising. There's some information on the swamp rose mallow distribution, how the stem count has changed over time, and the area of the extent that swamp rose mallow has been observed in. And this can be shown again in a nice animation showing how that area has really increased, especially over the Old Creek, where we had a lot of herbicide treatment take place. And Canadian Wildlife Service also did a really nice change detection showing the difference in frag and fish habitat over time. And I think both this table with the numbers and also this map really is telling by 2022 just how much more fish habitat we have available in these areas as a result of managing the frag. And then we move into our barn swallows. So the aerial insectivores, we see an increased abundance not only in the uninvaded marsh, but also in the areas that we treated with herbicide, which is awesome. And a little information about the importance of wildlife and how the recovery of that is taking place. So that's about it for the story map. We're hoping to get this all finalized and published. And I think it's going to be hosted on the Waterloo Wetland Lab website. So you can keep a lookout for that.
Wow, that's phenomenal. That's just a great product to have. Thank you so much for your efforts to pull that all together. How exciting. Really to see that too. It's really neat to have that the images before and after, just exactly the same photo point too. Just see the
Yes,
transition
yeah,
and along with all the
definitely.
data, but all the key components that have been looked at over the years as well in one place for folks to go in and have a look at. So thanks for your work on that, Grace. That's really exciting. And all of your compatriots that have contributed as well to that
Yes.
storyboard. That's really cool. Yeah. Thank you, thank you. I don't see any questions for you right now. I might be missing them. Help me out here, Gabby. From Lynn, short. Awesome story map. Love the before now. Just echoing what I had said as well. Yeah, it's really neat. So it's going to be on your labs access to looking at it, be on your labs website.
I believe so, yes. Yeah.
Okay, super. Thank you so much.
Great. Well, thank you very much for the speakers in that research panel. Really interesting and a really positive note to end on there. So thanks so much, Grace. We're going to take a really quick break, about 4 minutes here before jumping into our next session on Ontario Phragmites Action Update. So we'll see you there at the next session. Thanks, everyone.